Friends, today we will learn about Pseudomonas, stenotrophomonas, burkholderia. So, here we will study about morphology, culture, pigment production, biochemical reactions, resistance, antigenic structure, toxins and enzymes, and then typing methods, pathogenesis, laboratory diagnosis, treatment, control, epidemiology, and next we'll study about other pseudomonas. Then we'll study about stenotrophomonas, maltophila. Then we'll study about burkholderia malay or Pseudomonas malay, its morphology, culture, animal pathogenicity, human pathogenicity, laboratory diagnosis. Next, we will study about burkholderia, pseudomali, that is pseudomali, its morphology, culture, biochemical reactions, toxins, pathogenesis, laboratory diagnosis. Next, we will study about burkholderia cepacia. Next, here we will study about morphology, culture, pathogenicity. Next, we will study about non-fermenters other than Pseudomonas and burkholderia. So, let's begin. The genus Pseudomonas comprises of aerobic, gram-negative, non-formative, non-sporing, oxidase-positive bacilli which are motile, bipolar, flagella. Many species produce water-soluble pigments which diffuse through the culture medium. Majority of them are saprophytic, being found in soil, water, sewage, or wherever decomposing organic matter is found. The genus Pseudomonas belongs to the family Pseudomonaceae, which contains over 200 species. Human disease has been caused by P. aeruginosa, P.S. maltophila, P.S. malay, P.S. pseudomali, and P.S. capacii. The most important among these P.S. aeruginosa. Recently, P. malay, P.S. malay, P.S. pseudomalay, and P.S. cepacia have been assigned a new genus Burkholderia, which also belongs to family Pseudomonaceae. New names for this species are Burkholderia malay, B. pseudomalay, and B. cepacia. B. malay is non-motile. Pseudomonas maltophila has been named as stenotrophomonas maltophila, differentiating features of common encountered species. Now, let's know about Pseudomonas aeruginosa. Its morphology. Let's study about its morphology. It is slender, gram-negative bacillus, 1.5 to 3 micrometer into 0.5 micrometer, non-capsulated, non-sporing, and is actively motile by a polar flagellum. Most strains possesses pili. It is non-capsulated through mucoid strains may sometimes occur. Occasionally, strains have 2 or 3 polar flagella. Now, let's study about culture. It is a strict aerobe and grows well on ordinary media like nutrient broth and nutrient agar. The optimum temperature for growth is 37 degrees centigrade, but growth occurs at a wide range of temperature 5 degrees centigrade to 42 degrees centigrade. Now let's know about the nutrient agar. Colonies are smooth, large, translucent, low convex, 2 to 4 mm in diameter. The organism produces a sweetish aromatic odor. It is due to the production of 2 amino acetophenone. There are greenish blue pigment which diffuses into the medium. Now let's know about the Blood agar. Colony characters are similar to those on nutrient agar. Many strains are hemolytic on blood agar. Now let's know about the McConkey agar. Colonies are pale or colorless non-lactose fermenters NLF. And now let's study about citramide agar. It is a selective medium for PS aeruginosa. Peptone water. Let's know about the peptone water. It forms a turbidity with a surface pellicle. Pseudomonas being a strict aerobe tends to collect at the surface for more oxygen, hence forming surface pellicle. Now let's study about pigment production. PS aeruginosa produces a number of pigments which diffuse into surrounding medium. These pigments are pyocyanin. It is a bluish green phenazine pigment soluble in chloroform and water. It is not produced by other species of the genus, hence it is diagnostic of PS aeruginosa. Now let's study about fluorescence. Fluorescent, it is a greenish yellow pigment insoluble in chloroform but soluble in water. It may be produced by many other species also. Pyorubin, it is a reddish brown pigment that is insoluble in chloroform but soluble in water. Now let's know about pyomelanin. It is a brown in black pigment and is chemically unrelated to melanin. Its production is uncommon. Now let's study about biochemical reactions. PS aeruginosa derives energy from carbohydrates by oxidative breakdown rather than a fermentative metabolism. Special OF medium of Hughes and Leffinson must be used to find out oxidative metabolism. It utilizes only glucose oxidatively with acid production. Lactose and maltose are not utilized. All strains of PS aeruginosa and are oxidase positive and utilize citrate as the sole source of carbon. They are catalase positive and indole MRVP and H2S taste negative. They reduce nitrates to nitrites and further to gaseous nitrogen. Arginine arginine dihydrolase test is positive. Important biochemical reactions. Now we will study. Let's study about resistance. It is killed by heating at 55 degrees centigrade for 1 hour. It is resistant to the chemical disinfectants and can even grow in certain antiseptics like quaternary ammonium compounds, chlorosinolone, and hexachlorophene. Indeed, selective media have been devised for isolation of PS aeruginosa incorporating Dettol or cetrimide. It is sensitive to 2% aqueous alkaline solution of glutaraldehyde and also to silver salts. Due to its sensitivity to silver salts, silver sulfonamide compounds has been applied to topical cream in burns. It is intrinsically resistant to commonly used antibiotics. Examples of clinically effective antibiotics are polymyxin B, colistin, piperacillin, ticarcillin, cefotaxime, gentamicin, azlocillin, tobramycin, and ciprofloxacin. Now let's study about antigenic structure. O antigens. It possesses 19 distinct group specific O antigens. These antigens are heat stable. And now we'll study about H antigen. Two heat label H antigens have been recognized in PS aeruginosa. Now let's study about toxins and enzymes. Several toxins and enzymes produced by PS aeruginosa contribute to enhance its virulence. Now let's study about extracellular products. Pyocyanin inhibits mitochondrial enzymes in mammalian tissue and causes disruption and cessation of ciliary beat on ciliated nasal epithelium. Thus, this favors colonization of these organisms in the nasal mucosa. Now, let's study about extracellular enzymes and hemolysins. It produces proteases, general protease, alkaline protease, and elastase, hemolysins, phospholipase C, and heat stable rhamnolipid and lipase. This plays, this plays a key role in producing local lesions. Now let's study about exotoxin. It produces two exotoxins, A and S. Exotoxin A is a polypeptide of molecular weight 66,000 to 72,000 and inhibits protein synthesis. Mechanism of action of exotoxin A is similar to that of diphtheria toxin. Now let's study about endotoxins. It is a lipopolysaccharide, in short LPS, exhibiting many biological properties of enterobacterial LPS including pyrogenic action. Now let's study about typing methods. As PS aeruginosa is an important cause of hospital acquired infections, it is essential to type the strain for epidemiological study. Now let's study about bacteriocin typing or pyocin typing. Three types of bacteriocin or pyocins are produced by PS aeruginosa. These pyocins are known as R, F, and S. Pyocin producing strains are resistant to their own pyocins, though they may be sensitive to pyocins produced by other strains. Pyocin produced by the test strain is important to assess the growth inhibition of 13 indicator strains of PS aeruginosa. Depending upon the growth inhibition of these 13 indicator strains, 105 types of recognized. Pyocin typing is the most popular method employed for typing of PS aeruginosa. Now let's study about phage typing. Considerable difficulties have been encountered in bacteriophage typing. Now let's study about serotyping. Based on O and H antigens, 17 serotypes of PS aeruginosa are recognized. It is said to be reliable, but facilities exist only in reference laboratories. Molecular method. Let's study about molecular method. Restriction endonuclease typing with pulsed field gel electrophoresis is the most reliable typing method. Now let's study about pathogenesis. It is the most important antigen causing in nosocomial infections. Now let's study about pathogenesis. It is the most important agent causing nosocomial infections. It is due to its resistance to common antibiotics and antiseptics that is establishes itself widely in hospitals. Equipment such as respirators and endoscopes, articles such as bedpans and antiseptic or disinfectant solutions may be frequently contaminated. The other common infections by it are urinary tract infections following catheterization. Second point is acute purulent meningitis. Second point is acute purulent meningitis following lumbar puncture. Next point is third point is post tracheostomy pulmonary infection. Fourth point is septicemia in patients who are debilitated due to malignancy or immunosuppressive therapy. Fifth point is wound and bone infections. Sixth is chronic otitis media and otitis externa. Seventh is eye infections. Eighth is acute necrotizing vasculitis, which leads to hemorrhagic infection of skin and internal organs. Ninth point is infantile diarrhea. In the tropical areas, PS aeruginosa has been found to be responsible for a self-limited febrile illness resembling typhoid fever. In spite of its lack of invasiveness, PS aeruginosa does cause severe disease. The mechanisms of pathogenesis are not clearly understood, but it has been claimed that pathogenical processes seen in infection are caused by exotoxins, proteases, elastases, hemolysins, lipases, and enterotoxins. Exotoxin A is a lethal toxin acting like the diphtheria toxin. Elastases, the organisms produce blue pus, and the term arginosa, meaning bhordy grease, which is bluish green in color, and biocyane being a literal translation of blue pus. Now let's study about laboratory diagnosis. First is specimens. Pus, wound swab, urine, sputum, blood or CSF. Now let's study about culture. Specimens may be inoculated on nutrient agar, blood agar or McConkey's agar and incubated at 37 degrees centigrade for 18 to 24 hours. On nutrient agar there is bluish green pigment diffused in the medium. On McConkey agar they grow as a pale colonies. That means N in short NLF. In peptone water, surface pellicle and green pigment can observed, and selective media such as centrimide agar may be necessary to isolate PS aeruginosa from fishes or other samples with mixed contaminant. Isolation of the bacillus from a specimen should not always be taken as etiological agent. Repeated isolations would help to confirm the diagnosis. Now let's study about gram staining and motility. They are gram-negative bacilli and are actively motile. Now let's study about biochemical reactions. The oxidase test is positive within 30 seconds. They are non-fermenter. They break down glucose oxidatively with acid production only. Other biochemical reactions also help to confirm the isolate. Now let's study about typing method. Piocin typing is the most commonly used method. It is mainly used for epidemiological studies. Now let's study about antibiotic sensitivity test. It is useful to select out proper antibiotic as multiple resistance to antibiotics is quite common in PS aeruginosa. Now let's study about treatment. It is intrinsically resistant to most of the commonly used antimicrobial agents. Ciprofloxacin, piper, piperacillin, ticarcillin, azlocillin, cefotaxime, septazidime, then gentamicin and tobramycin are used in treatment of PS aeruginosa infections. Now let's know about control. Cross infections in hospital are to be prevented by constant vigilance and strict attention to asepsis. A polyvalent vaccine made from the cell surface of 17 recognized serotypes of PS aeruginosa has been claimed to stimulate active immunity in man. Now let's study about epidemiology. Cross infection. PS aeruginosa is a saprophyte and its ability to persist and multiply in most moist environment of hospital wards, kitchens, equipments and antiseptics or disinfectant solutions is of particular importance in cross infection. Now let's study about epidemics. Epidemics and outbreaks of PS aeruginosa infection have occurred among newborn nurseries and young infants in pediatric wards. Now let's study about risk group. These include burn patients, patients with immunosuppression and patients with patients who have undergone cardiac and renal surgery. Now let's study about other Pseudomonas. PS putida, PS stutzeri, and PS fluorescens and becoming clinically relevant in increasing percentages. Now let's study about stenotrophomonas maltophilia, or we can say Pseudomonas maltophilia. This is an opportunistic pathogen causing wound infection, urinary tract infection, and septicemia. It is oxidase negative. It acidifies maltose in addition to glucose. Now let's study about burkholderia mallet or Pseudomonas mallet. The bacillus was first isolated by Lofer and Sears in 1882 from horse dung of glanders. It causes a natural disease called glanders in animals, horses, mules, and asses, and man occasionally acquires the infection from animals. Now let's study about morphology. It is a small 2.5 micrometer into 0.5 micrometer non-motile gram-negative bacillus, often giving a beaded appearance. Now let's study about the culture. It is an aerobe and facultative anaerobe, grows on ordinary culture media. Colonies are small and translucent. Initially become yellowish and opaque on aging. Now let's study about animal pathogenicity. In susceptible animals like horses, mules, and asses, B. Mali produces two types of lesions, glanders and farcy. First is glandar. Let's know about the glandar. In glandar, the respiratory system is affected and the animal develops profuse catarrhal discharge from the nose. The nodule formation starts in the nasal septum and later the nodules break down to form ulcers. Now, let's study about farcy. Farcy follows infection through the skin with involvement of superficial lymph vessels and lymph nodes. The lymph vessels become thickened and stand out as hard cords under the skin, which are called farcy pipes. Intraperitoneal inoculation of bacilli in male guinea pig causes testicular swelling in 2 to 3 days due to inflammation of tunica vaginalis. This is known as Strauss reaction. This may be followed by death of the animal in 1 to 2 weeks. The Strauss reaction is not diagnostic of glanders because similar reactions in guinea pig may also be observed by Brucella species, Pris, Noccard bacillus, Actinobacillus, and B. Pseudomonas. And now let's study about human pathogenicity. Humans may become infected via skin abrasions or wounds through contact with discharge of a sick animal. Human disease may present as acute fulminant febrile illness or a chronic indolent infection producing abscesses in respiratory tract or skin. The fatality rate in glanders is high. The laboratory cultures are highly infectious to work with. Now let's study about laboratory diagnosis. First is specimens. Purulent discharge from the lesion. Second is direct microscopy. Gram negative gram negative beaded bacilli are found on gram staining of the specimen. Next point is culture. Third is culture. Culture is done on blood agar. Next is animal inoculation test. It is done in guinea pigs. Strauss reaction can be demonstrated. Now let's study about allergy test. Malin test. First is Malin test. Malin is a preparation from the organism bee malin and is analogous to tuberculoprotein. Intracutaneous or subcutaneous injection or malin evokes a delayed hypersensitivity reaction in animals suffering from glanders. Now let's study about burkholderia pseudomali, that means Pseudomonas pseudomali. It was formerly known as Whitmore's bacillus, actinobacillus, Whitmore myces, pseudomali Loifleral pseudomali. It is the causative agent of malioidosis, which is a glanders-like disease and epizootic in rodents in Southeast Asia. The name malioidosis is derived from melis, a disease of horses, and idosis, widows meaning resemblance. The organism was first described by Whitmore and Krishna Swami in 1912. From a glanders-like disease from a man in Rangoon. Now let's study about the morphology. It resembles bee mallee but differs in being motile. Next is culture. It is similar to bee mallee and let's know about the biochemical reaction. It resembles bee mallee but differs in being liquefying gelatin and forming acid from several sugars. Now let's study about the toxins. Two thermolabile exotoxins, one lethal and other necrotizing, have been identified in culture filters. Now let's study about pathogenesis. B. pseudomolai is a saprophyte of soil and water with a large animal reservoir. Melioidosis occurs in rats, rabbits, and guinea pigs. Human infection may occur by inhalation or through skin abrasions or by arthropod vector. Disease is epidemic in Southeast Asia. Subclinical infections are common in man. There may be an acute septicemia, a subacute typhoid-like disease, or pseudomonia and hemoptysis resembling tuberculosis. Acute disease has a high case fatality rate. In chronic form, the organism may localize in any tissue producing caseous necrosis or suppurative lesion. In India, cases of melioidosis have been reported from Maharashtra, Tamil Nadu, Orissa, Kerala, Tripura, and West Bengal. Now let's study about laboratory diagnosis. The organisms may be detected as small, typical bipolar septic spin appearance with methylene blue stain. Small irregular stains, gram-negative bacilli in exudates may be isolated from sputum, pus, urine, or blood. Antibody to P. pseudomoli may also be detected in patient serum by ELISA and indirect hemagglutination assay, in short IHA. Now let's study about burkholderia cephacea or Pseudomonas cephacea. Let's study about the morphology. It is a slender gram-negative bacillus which is motile by multitrichous flagella. Bacillus accumulates poly-beta hydroxybutyrate as granules, hence stains irregularly. Now let's study about the culture. It is aerobic and grows well on ordinary nutrient agar optimally at 25 to 35 degrees centigrades. Colonies appear in 48 hours. Most stains do not grow on DCA medium. Cultures on blood agar die in 3 to 4 days. On prolonged incubation, colonies become reddish purple due to formation of a non-diffusible phenazine pigment. Now let's study about pathogenicity. It is a low-grade human pathogen and an important cause of nosocomial infection. It is being recognized as an opportunistic environmental pathogen, particularly in those with cystic fibrosis, in whom it causes fatal necrotizing pneumonia. It is resistant to a large number of antibiotics and contaminate antiseptic solutions, including chlorhexidine and cetrimide. It can also survive in distilled water up to a year. Now let's study about non-fermenters other than Pseudomonas and burkholderia. Several gram-negative bacilli may be non-fermenters of carbohydrates and may be confused with Pseudomonas or burkholderia species. Some of these non-fermenters are Actinobacter, Alkaligenes, Achromobacter, Flavobacterium, and Echinella species. Finally finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.